So thank you back, everybody. I hope you had a good break, short but good, time for some food. So I am very happy to help Wynand to co-host the British session this afternoon. It's my pleasure to introduce the first contributed presentation that will be by Yael Grosjean. Yael is a group leader at the Center for Taste, Smell and Feeding Behavior in Dijon, in France, and is going to talk about adult sex-specific vigilance via olfactory receptor inhibition in Drosophila. Yael, the floor is for you. If you can share your screen.
Okay, so as I said, I will talk about some data we obtained the past few years in the team about some effects of volatile molecules or odors in Drosophila and what are the consequences of these odors and how to at least explain a part of this effect. And so, as you know, olfaction is very important to detect danger, to find some food, to communicate between individuals, and even to find a mate for insects. So they play a very key role in their ecological and life. And understanding how these odors could modulate behavior and what are their impact is highly relevant. And some effects of these volatile molecules could lead to reversible anesthesia in Drosophila. This is the typical case of CO2, for example, to anesthetize insects and of course here on the picture some Drosophila to collect them and to manipulate them. So these volatile molecules could impact a lot the physiology and the nervous system activity. And by screening the effect of some odor, we identified that two of them, propanoic acid and butyric acid, are able to anesthetize Drosophila. And especially at a concentration of 2%. At a lower concentration, there is no anesthesia. At 2%, there is an anesthesia. And there is no anesthesia for a similar compound with similar volatility, that is acetic acid. And we screen several other acids from C2 to C10 to see if at 2% there was some similar effect compared to propanoic and butyric acid, and it was not the case. So only butyric and propanoic can lead to a reversible anesthesia. Of course, if you remove the odor, they wake up. And we also tested an amine, pyrrolidine, that is a known repellent amine, to be sure that at 2%, this is not a general effect of repellents that could cause this anesthesia. And this is not the case, again, at 2%. And we compare also the effect of these propanoic. So first, to continue the talk, I will concentrate only on propanoic acid to be much simpler, but we have also data for butyric acid. So for propanoic acid, we compare the effect to CO2 and NO anesthesia, so nitric oxide anesthesia. And all of them are reversible anesthesia with these volatile molecules, but they lead to various effects on the muscular tonus. For example, with CO2 and NO, you have the legs that are contracted, you have the wings that could be a little bit extended. And for propanoic acid, this is never the case. The wings are totally fine, close to the back of the fly, and the wings are relaxed in a normal position, but the fly is on the side or on the back because they got anesthetized. So there are different effects on muscle tonus with these odors. And what is even more interesting and puzzling is that the effect of propanoic acid is totally different in larvae. So again, in adults, there is an anesthesia with propanoic acid, plus propanoic acid is also repulsive for Drosophila adults. But in larvae, the effect is totally opposite. These molecules, propanoic but also butyric, are attractive molecules for larvae, and they never cause anesthesia. Even at 100% propanoic acid or butyric acid, that's the same, there is never any anesthesia in Drosophila larvae. So it means there is something specific in adults that is causing this sensitivity and this anesthesia. And of course, we control that larvae are able to be anesthetized by CO2 and NO, and this is the case. It takes a few seconds to anesthetize larvae with both CO2 or NO, and it takes a little while, a little bit more time to recover for CO2 anesthesia compared to NO in larvae, but these are details. And the effect is very strong and very consistent. So there is definitely a different sensitivity between CO2, NO, and propanoic acid, and of course butyric acid, in Drosophila, and the effect again is highly specific for adults. And what is even more interesting is that the effect is different between females and males at 2% propanoic acid again. So it takes less time to anesthetize males compared to females. So it takes more time to anesthetize females. And we could think, okay, that's because they are bigger. And in fact, we have several clues to say that this is not due to the size of the fly, that we have this effect and the different effect between male and females. And first, so we wanted to know where this difference in anesthesia is coming from. Is it a part of the head that is causing this? And our idea was maybe this is coming from the antenna, the olfactory organ of the fly. And indeed, this is the case. If we decapitate the flies, then the males are behaving as females. So it takes more time to anesthetize these males. So again, this is an indication that this is not due to the size of the fly. So, okay, nice. So as I said, so the head is playing a role in the gender sensitivity to propanoic acid at 2% to get anesthetized. And as I said, our main hypothesis is that this effect could come maybe from the antenna. So to test that, we took advantage of the genetic tools available in Drosophila, and we have several null mutants to test the different olfactory families of olfactory receptor and to inactivate a wide range of olfactory receptors, so to target the co-receptors. We targeted 3 different co-receptors to screen all the specific receptors. And first, we tried to see if this is coming from an IR80 co-receptor, olfactory neuron expressing this co-receptor, because they are known to detect acids. And to our surprise, males are still behaving as males and females the same. So then we looked at ORCO, null mutant, and then the difference between male and females disappeared. So it seems that this effect, the different sensitivity between male and females, is coming from ORCO and is coming from a specific OR receptor associated with this co-receptor. And we have a control with a different a co-receptor that is IR25A targeting amines, mostly amines. And so then, to confirm that ORCO is really important for the male response, we used the ORCO-GAL4 to express in ORCO-expressing sensory neurons the Shibire thermosensitive isoform to block the activity of these neurons. And when we do that with the thermosensitive forms of Shibire, we could show that we could transform these males to females. There is an increase of the delay to respond to propanoic acid, so it takes more time for the males to get anesthetized. And these are the controls then. We did the same with the females, there is no effect. So male sensitivity through ORCO neurons. We then investigated if the difference of sensitivity in ORCO expressing neurons is related to a pathway associated with a male transcription factor responsible to the differentiation in a male olfactory neuron that is fruitless. And when we use the fruitless null mutant, then we transform the males into a female. So this seems that this is coming from fruitless. We tested the two different OR associated with fruitless and found that when we use a null mutant for one of the olfactory receptor expressed in fruitless neurons and in ORCO neurons, we could transform OR males into females, and this is OR47B null mutant. We confirmed this using the same OR47B GAL4 driver associated with Shibire-TS, and we could thermosensitivity in a thermosensitivity manner transform the male into a female in a transient manner. So it takes more time when we inactivate these neurons in males. So male sensitivity is coming from OR47B. So we then used the genetic tools to perform some calcium imaging. We started some years ago, and we started this experiment with US GCAM3, and it worked. So we continued with this marker. So this is the glomerulus expressing, so the glomerulus, the target of olfactory neurons expressing OR47B, this is DA1B, here, located there. And this is our picture with the calcium imaging. So these neurons have a basal activity and they're fluorescent. And from the work of the Carlson lab, we were already aware that propanoic acid is able to inactivate OR47B expressing neurons. So we were expecting a decrease in activity of these neurons when we applied propanoic acid. And indeed, this is what happened. And let me start.
3 minutes left.
Okay, that should be fine. So this is, you see the pulses when we apply propanoic acid, there is a decrease of the activity of these neurons. We quantify this, and we have, here we have a mix of male and females, and we see a nice dose response to propanoic acid to inactivate these glomerulus. So then we separated male and females, and we found that at 1 to 10%, we have a gradual response in females. Instead, in males, where we have already non-significant difference at 1% compared to 10%. So it seems that they are more reactive to propanoic acid, males again. We use a null mutant, and this response is disappearing. So this is indicating that the inhibition is really coming from OR47B neurons and not from a lateral inhibition of these glomerulus. So OR47B is specific for the gender inhibition in male compared to female. leading maybe to the anesthesia of the flies. And then we looked at different controls. So first we tried to be sure with genetic tools that the taste organs are not involved, and this is the case. So this is the poxen mutant and rescued everywhere, but not in the taste organs. So the taste organs are transformed into mechanosensory organs. And so when you have no taste, you have still the wild type response. And then we were looking at what happened in other tissues to explain maybe why there is an anesthesia, and we used the Yael driver expressed in trachea. And when we block the activity of the cells expressed in the trachea, we could delay the response to propanoic acid, so it takes more time to get anesthetized. But this delay is still present in male and female, and you have still the difference between male and female. This is the middle bars. And if we decapitate these flies, of course, we lose this response. And we confirm this. We have neurons close to the trachea, so trachea may be involved in the sensitivity to propanoic acid. Finally, we showed that volatile molecules, so propanoic acid, has no gender effect in closely related species, Drosophila suzuki. And now you have no difference between male and female. We have more data on other insects, but I have no time to present.
Time is over.
Okay, and this is the end, just to show the people that have produced the data. And if people are interested in a postdoc in the lab, they are welcome. Thank you very much.
Thank you. So is there any question? I don't see any question in the Q&A. So there is no question from students. So maybe I have one question for you. What could be the ecological significance of adult sensitivity to anesthesia and not to larvae?
What I could imagine is that larvae are in the food and in decaying fruit, so they cannot escape a lot from the area where they are feeding. And most often for Drosophila, the fruits are again decaying, so they have lots of complicated molecules that could be toxic, so they need to be resistant. And the adults, they maybe are more picky for the odors that they smell, because they use their olfactory perception for lots of different reasons, and they need to fly in different areas and recognize dangers and so on. So maybe this is why they need a more accurate effect compared to larvae. That's a possibility.
Okay. Thank you. Is there any other question? I
see a question, but I don't know.
No.
No.
So if no more question, we can move
to
the next speaker. So I'm happy to welcome the next contributed presentation. It will be by Mario Pannunzi. Mario is a postdoc fellow at the University of Sussex in the United Kingdom, and he will present his work on non-synaptic interactions in olfactory receptors neurons, a peripheral odor coding. So Mario, if you are ready, you can share your screen.
Thanks. Okay. So before starting, can you see? Yes, I hope so. Before starting,
Yes,
I would like to thank the Levium Trust, the University of Sussex, for funding this research and the one that I'm researching now. And of course, the organizer of the Insect Olfaction and Taste for organizing this very nice conference, really spot on for us. But most of all, I would like to thank Thomas Nowotny, the person that really believed on this research and on me on doing it. So let's start with this nice effect of non-synaptic interaction. So typically, the neurons are not interacting between each other because they are isolated with the myelin. And this happens for almost all neurons. And there is a parallel for the cable that we have in our houses that are shielding. And of course, we don't want this kind of interaction, electromagnetic interaction, both on neurons or on cable. But there are some exceptions, and this is one of the that we want to talk about. So here we are looking at the results and the figure from the very nice paper from Su et al., where they analyzed the Drosophila model. And in the Drosophila, typically the olfactory receptor neurons are co-housed on the sensillum in group of 2 and 4. And they show that presenting a first odor stimulate the first olfactory receptor neuron, and then presenting a second odor will stimulate the second one. So in this case, AB3A and then AB3B, that will inhibit the first olfactory receptor neuron. So this is kind of strange because, of course, we can interpret it like a disruption of the signal. So why it could happen and why we should see this effect? Of course, we would like to see some kind of function in it, or we like to understand if it is just a spandrel that doesn't cause anything. To do that, we analyze the function of the olfaction. And typically we think of olfaction to identify odor or the intensity of the odor, and or the ratio between the concentration, or even to identify the location of the source. And in particular, what we were interested on are 3 hypotheses. The first is that NSI, so this interaction, can help to encode the concentration ratio, or to encode the correlation between concentration, or finally, it could increase the dynamic range. In this talk, I would like to explain you these 3 hypotheses, the intuition behind them, and how we model the early sensory, olfactory sensory area of the Drosophila, and show some result about it. So let's start with the first hypothesis. If we think of a very simple system with 2 olfactory receptor neurons and 2 odorants that are stimulating these neurons, and we think of spanning different level of concentration for one odorant, we are used to see this dose response curve. And if we kept fixed the other odor concentration, of course, we can imagine that the concentration ratio is not perfectly encoded by the frequent firing rate ratio at the level of the photoreceptor neuron or the trajectory neuron that are in the antennal lobe. But the NSI, this interaction, can decrease the activity of the second neuron. Here we can see on this diagram, and therefore straighten up this curve that represents the firing rate ratio. In this way, the NSI could help to encode the concentration ratio. And this is the first hypothesis. For the second one, we need 2 parts of information. First, that the insects in general can use and can understand whether an odor is is coming from two others, are coming from the same source or from different sources. Here there was this nice demonstration from Silke et al. Where they show that the MOF can even decode 1 millimeter distance between the source of the odorants. In that case, there was a pheromone and an antagonist of the pheromones of the MOF. And the other part of information is that when odorants are coming from a same source, are much more correlated than when they are coming from separated sources. This is well illustrated by this experiment. With these two informations, we can imagine that NSI can help to encode the correlation between two odorants. So, for example, if two odorants are coming from the same source, their concentration are correlated, and therefore the NSI are more active, and therefore the activity of the response is less. The opposite happens when the odorants are separated from the beginning, therefore the correlation is less, and even less is the NSI activity, and therefore the activity of the oral ends is untouched. And this is the second hypothesis. For the third one, we can imagine again the two olfactory receptors and how they typically respond with a dose response with the ceiling effect. The ceiling effect, of course, disrupts the information of the intensity. And maybe the NSI can decrease this ceiling effect and increasing in this way the dynamic range. The dynamic range is the distance between the first detection and the last encoded clearly the intensity of the concentration. So this is the third one. What we try to do is a model of the Drosophila olfactory system. Of course, we know that the Drosophila has more than 60 different receptors, all stereotypically co-housed on this sensilla, and they are connected one-to-one to the antennal lobe of on the trajectory neurons that are inhibited by the lateral neurons. This is, of course, a very raw description of the olfactory system, as we have seen in these days, couple of days, how complicated it can be. But to do this model, we even need to simplify further. And in this case, we are using just a single sensillum with two types of receptors that are connected with the two types of trajectory neurons and lateral neurons. And we have done, of course, many different assumptions. And for example, the interaction between the neurons at the NSI is implemented through the reversal potential, or that the neurons are leaking integrated fire. This is our model, and we constrain the model with different observed effects in the literature. For example, from Martelli et al., we know that the intensity invariant shape of the oral ends response to a single step, or the shape we simulated, like in Leal et al., responding to a single step, to a ramp, or to a parabola, or the function connecting trajectory neurons response to the olfactory receptor neurons shown by Olsen and Wilson. These are our constraints. To test our first hypothesis, we used three different models. The control model, where the two types of receptors are not interacting at all. The LN model, where they are interacting only at the level of the antennal lobe and not through the olfactory receptor neurons. And the NSI model, where the interaction happens at the level of the olfactory receptor neurons. And we analyze this system with the single pulses of different duration and concentration. We analyze the response, so the firing rate of the trajectory neurons, olfactory receptor neuron. We observed that increasing the concentration ratio and for different level of concentration, for the control model, so on the left, we can see that we cannot have a good encoding in the ratio of the trajectory neurons of the concentration ratio. The LN model helps a bit, but not much, while the NSI model looks like it's straightening up these curves and therefore helping quite a lot on encoding the concentration ratio. For the second hypothesis, we have to simulate realistic plumes. And we have done using the statistics observed in literature about the concentration, the wave duration, and the blank duration. And we stimulated the model with a different level of correlation between two concentrations, between two odorants. And of course, for the we used again the three models shown before. And for the control model, there is no effect at all for the correlation. So we can see on this diagram. But both for the NSI model and the LN model, we have an effect changing from the zero correlation to very high correlation. Of course, we would like that for a good encoding that at zero correlation, the interaction doesn't affect much, because otherwise it will be a disruption. Whilst we want a high effect on a very high correlation. And you can see that it is true for the NSI model more than for the LN model. And finally, for the third hypothesis, we analyzed the system using the response of two different olfactory receptor neurons that have a slightly overlap on the response of this odorant, for example. And we analyzed the dynamic range, so the response from the 10% of the first oral ends until the 90% of the second oral end. That is what is shown in the diagram on the top. And we analyze it for different level of similarity between the oral ends. And we can see on this diagram on the bottom that the control is approximately similar to the single oral ends dynamic range when they are really similar. And this is quite obvious, no? Because it's very similar to a single oral end, and it increases for increasing similarity. But the NSI in this case is not helping. On the contrary, it is decreasing the dynamic range when they are very similar. And this is on the opposite of our hypothesis. So to recap,
5 minutes.
Yep, thanks. To recap, we have these three hypotheses. The first hypothesis was, the first and the second hypothesis was confirmed, but the third one was not. And as we consider interesting the second hypothesis and very clear, and we have a good idea to how to test it so that improve the concentration correlation with Thomas Nowotny, we wrote a project and tried to test it. And to do that, we kind of have a lot of collaboration, luckily in the University of Sachse. So thanks to George Jimenez, that is an expert on electrophysiology, Claudio Alonso, a Drosophila expert, and with a supervision from far away of Paul Schiska, we have done this group. Now we have even the very good help from the PhD students of Lydia and the technical help of Andrei Mayachasas. And with this team, we are trying to do this project. That is an interaction between electrophysiology, behavior, and the model itself. We would like to test the fact that for different location, we have an improvement of, thanks to the NSI of the encoding. And we would like to use the Reaper and the TNT to check this. The Reaper, of course, we will remove completely one of the neuron and therefore remove completely the interaction. While the TNT will be used, the TNT GALP4, to remove the effect coming from the neuron to the antennal lobe. This is our idea, and we are already started the project and working for it. And finally, we will do it at the final test on a wind tunnel and compare the response from these two genes from these two fly lines. And that's it. Thanks. Maybe I finished a little bit early. Yep, I concluded.
Yes, thank you very much. Thank you for this very nice and interesting talk. There are some questions. So I will read a question from Martin Navrot. You tested two models exclusively, peripheral non-synaptic interaction versus lateral inhibition in the antennal lobe. What do you expect when combining the two models, since both mechanisms seem in place?
Yes, absolutely. This is a quite good question. We first of all, we tested the two models separately because we would like to test this hypothesis, that is, are the NSIs useful in some way or not? And therefore we wanted to test this separately for a reason, that is, avoid interaction to have a clear response. But of course, the nature is much more complex than our model, and typically the two elements are together. So we expect something in between. And of course, I expect that depending on which ORNN or which sensillum we are looking at, they can happen intermediate level. So for example, at the level of the pheromones, we can expect the NSI can be more specific. So we can be quite at the level of where the second hypothesis is the most important, and maybe the interaction at the level of antennal lobe is less important. For other kind of ORNs, it can be more important other parts. Anyway, what we have seen is that in any case, the NSI can be helpful for both elements, and the LNs is slightly less important and is slightly disruptive at the level of the interaction. So having both of them together shouldn't be changing too much. But right in this moment, we are running the simulation for understanding it. So probably we'll have a complete answer to this soon.
Okay, thank you. There is another question from Alexander Steinbrecht. We still have time. How exactly is the non-synaptic interaction in your model?
So there is, in the literature, there is a nice analysis of the fact that this interaction should be happening at the level of electric level. So Zhang et al. Have done this experiment where an electrode passed through two different sensilla, and in this way they have an interaction. Now we consider the various hypotheses, but we don't have enough data to be sure. What we have done is to connect the input from the olfactory receptors to the activity of directly of the reversal potential. So manipulating this reversal potential is affecting the activity of the other neuron. I hope that I answer. Yeah, so, in bArchive, our it's allowing our paper, so we can discuss all the different equations. I hope that is enough for now.
Okay, thank you. So I don't see any other question. Yes, one question. Non-synaptic inhibition occurs between a specific pair within one sensillum. Do you think the pairing has some meaning?
Yeah, that's exactly where all our research started from. So these, at least for the Drosophila, they are really stereotyped, and there are different papers on different lab illustrating that. So it could be really important at the level of the pheromones, and there are some results showing that it can disentangle the effect of mated female or not. Or there is even I just discovered this very nice paper where on ants, where they have even 200 different receptors and neurons on the same sensilla, where they can recognize whether the other ants is coming from the same nest or not, and therefore attacking it or not. So yes, we believe that there can be some reason. But of course, as illustrated by all these conferences, the system is so complicated for the olfaction that we're just speculating for now.
Okay, thank you very much. So I don't see any more question about your talk, but there is one technical question since we have 1 minute left. So there is someone that is trying to follow the workshop on YouTube, but is not able to see the very first talk. He's able just to go at hour 10, hour 12. So I cannot respond this technical problem. Maybe Walter or Coral or Wynand can solve this. I don't know. Maybe it will be fixed later. Sorry. Okay, so now we will move. Thank you very much again. And we will move to the next speaker. I will share my screen. Oops. So, how does it work? I cannot change. Do you see my screen? So I cannot
use the mouse.
Yes, it's what I am doing. yes, okay. So now it's again. No, yeah. Sorry. So the next speaker is a keynote lecture. So we have the pleasure to welcome Silke Sachse. Silke is a research group leader for leader of the independent group on olfactory coding at the Max Planck Institute of Chemical Ecology in Vienna. She's been leading this group since 2008. And the main research aim, her main research aim is to understand how neural circuits organize olfactory inputs to control behavior in insects. And she's using mainly Drosophila melanogaster as a model organism. She's combining neurogenetic tools with in vivo functional imaging and behavioral assays to elucidate the olfactory circuitry in the fly brain. And today she's going to talk about olfactory learning that modulates a neural circuit mediating innate odor-guided behavior in Drosophila. Thank you very much, Silke. I stop sharing my screen and the floor is for you if you are ready.
Thank you very much, Emmanuelle. It was a nice introduction. I'm sharing my screen. Okay, thank you very much for inviting me to give here a long plenary talk. It's really a very, very nice idea to put such a program together. It's fantastic. So thanks to Walter, Wynand and Karl. And today I will talk about mainly learning, but before I come to an ongoing study where I would like to show you some details, I would briefly like to tell you in general what we are doing. So I'm research group leader, the Max Planck Institute. My group is hosted by Bill Hanson's department. And what we are doing, our aim is really to really understand how the neuronal circuitry is organizing olfactory inputs to enable an animal to take odor-guided decisions, to enable an animal to find the right food side, right, to discriminate between this green and rather ripe banana here, to find and locate the right oviposition sites and not to oviposit on this mold apple here. And also on pheromone detection, of course, the olfactory system is essential. So our approach is always to link the neuronal circuitry to the behavioral output. And as one of the key techniques, we use 2-photon functional imaging to visualize how odors are represented in the brain at different levels. We also use a lot of anatomical methods to visualize neurons, to trace them, to find and identify their target areas. And on the behavioral side, we have different behavioral setups in the department. These I'm always doing mostly in collaboration with Markus Knaden, who is heading the insect behavior group in the department. And there we have just simple T-mazes for simple innate preferences, the fly walk, which has been invented by Markus Knaden. We are currently also developing a tethered flight setup so that we can also observe flies and measure brain activity while they are flying. Then we, of course, also study reproductive behavior and have courtship and oviposition assays. And since we are working with Drosophila, and since this is a genetic model organism, we, of course, use the genetic toolbox that's available and also generate new genetic tools to activate and silence selective neurons using optoneurogenetics and really to understand then the impact of single neurons or certain neuronal populations for odor-guided behavior. Before I start now with some projects, give you some insight what we have done and what we are currently doing, I would briefly mention also what Bill's department is doing. Bill Hanson, he's unfortunately not talking at the symposium. And his department, especially in the last years, has really extensively extended the scope and has really extended the number of species to work on and to understand odor-guided behavior. So one major or several projects are on sister species of Drosophila to understand what are the different strategies of flies that live and have adapted to special niches. One big project where I'm also involved is on locust, on the desert locust, and we are trying to really elucidate all the details of the olfactory pathway, really from the receptors up to higher brain centers and also up to behavior, because they have such a different anatomy than any other insect. And then in Bill's department, of course, there's also a big group working on moths with a special focus on Manduca sexta, but also other moths, and to understand their strategies to perform odor-guided decisions. Then there's another big thing that has happened 2 years ago about we established a Max Planck Center called Next Generation Insect Chemical Ecology, which Bill initiated, and that's a huge joint venture of our department together with SLU and Lund University. And in this Max Planck Center, we are aiming to study the effects of the Anthropocene. This means the effect of human activities on insect chemical communication, and to understand how really global changes in the environment, how do they affect odor-guided behavior and odor coding. And we are studying this in flies, in mosquitoes, in moths, in bark beetles, and we are studying the effects of ozone, carbon dioxide, temperature, and nitric oxide. Okay, now I'm coming back, switching back to Drosophila and to some past and current projects. I would like to give you some more introduction, so I walk you very briefly through the olfactory pathway, although that's probably not needed for this audience. But I would like to just introduce the key players in the system, which are the sensory neurons that innervate the first olfactory neuropulse, the antennal lobe, consisting of 50 glomeruli. There's a lot of processing taking place, which is accomplished by a huge diversity of local interneurones, which are mainly GABAergic, inhibitory. Then the olfactory information is relayed to higher processing centers. It's the mushroom body, center of learning and memory. Ilona talked about this morning, and the lateral horn, which is the center for innate behavior. Now, as I said already, we use two-photon functional imaging to study how odors are represented in the brain. And here you see some raw data movies from how odors are evoking, they always evoking bilateral symmetric pattern in the left and right antennal lobe, and it's always a combinatorial code that an odor is usually inducing. And we have generated an in vivo atlas of the antennal lobe to be able to assign the activities of glomeruli to specific glomeruli, to identify glomeruli, so that we can compare those patterns between different individuals. And that led us then to establish a functional atlas of the antennal lobe where we can show really the specific activity pattern for each odor. And this has been done for many, many species, and during the last 2 decades, many groups, including us, could show that odors are really coded by a combinatorial code, meaning that one odor is inducing pattern in a specific combination of glomeruli, while each glomeruli receives or can detect several odors. There are, of course, also the exceptional cases, which are the labeled line on the other side, which are pheromones. The pheromone is usually, one receptor is usually dedicated to detect a pheromone. And such a labeled line strategy has also been shown for geosmin, for carbon dioxide, and a few others. Now, summarizing, we have in the antennal lobe a combinatorial code which is encoding odor specificity. On top of this, we could show some years ago that there's also a valence code, meaning that the hedonic valence of an odor, whether an odor is pleasant or unpleasant to us or an animal, is encoded also in those patterns. This means if an odor is inducing approach behavior and is attractive, certain glomeruli are activated in this combinatorial code, which are shown here in blue. And when an odor induces repellent behavior, then these magenta glomeruli are activated. And this has been shown, this we showed a few years ago together with Markus Knaden, and it's published in Cell Reports. Now, in a natural environment, animals are not exposed only to single monomolecular odors, which are aversive or attractive or neutral. Sometimes they can also be exposed to a conflicting sensory input, conflicting environment, like in this case, where there is a food source, and on top of this food source, we have a lot of mold. So now the animal needs to decide, do I oviposit here, or will my offspring die in such an environment? And therefore, we studied how conflicting sensory input might be coded by the olfactory system. And what we did is we just combined odors which are attractive and aversive and represented them in the mixture, and then dissected the olfactory circuitry and the processing strategies of the circuitry. What we could show is that there is a lot of interaction taking place, and there is a specific crosstalk occurring in the antennal lobe in a way that the input channel, or those sensory neurons, and therefore those glomeruli that respond to repellent odor, that they're interacting with the input to the attractive odor in a way that the input of the repellent odor is suppressing and it's inhibiting the input to the attractive odor. That then leads to a decreased output of this attractive input channel, and therefore then leads to a reduced attraction. So if the concentration of the repellent odor is too high, it will just suppress the input to the attractive odor, and the animal won't approach and won't oviposit here anymore. And that's done by lateral inhibition. Now, odors do not need to be always conflicting. They can also be synergizing. They can also be reinforcing. And one example for such a case is pheromone detection and food odor recognition. We could show that food odor is really interacting with pheromone detection. So flies have food odors always in their environment, in their background, because they do everything on food. They meet on food, they aggregate on food, they court on food, they mate on food. So we therefore studied the impact of food odor on pheromone detection. And we could show that if where there's a food odor in the background, the food odor is boosting the input to the sex pheromone in virgin female flies. So virgin female flies is way more sensitive to the male emitted sex pheromone when there's food odor. And this, we dissected the mechanism and could show that this is done by electric coupling it's an electrical and a excitatory lateral excitation that's boosting the input to the sex pheromone. And this boosting then leads to a stronger output, and then it leads to an enhanced attraction. Meaning that the female virgin fly would start copulating with a male much earlier when food's around. And that's a pretty useful mechanism because it ensures for the female flies that there is a nutritional supply for her offspring later on when she's starting to oviposit after the copulation. So it shows, these two examples shows that a lot of processing is going on in the antennal lobe, really to encode complex and diverse odors that are occurring at the same time. Now I come to the main part of my talk, which is plasticity. So we understand more and more the underlying olfactory circuitry, the key players. We understand more and more how our odors represented in the system and how they are coded. But we're also wondering now, how plastic is this circuitry? How well can it adapt to, for example, a changing environment? And how well can it adapt to experience? So what we are doing in one project currently is to see what happens when you raise an animal in a different odor background. What we are doing is we are raising flies in the background of geosmin. Geosmin is a pretty well-defined neural circuit. It's the odor of mold. It's highly aversive, and it's detected only by a single class of receptors, odorant receptors, and therefore it's detected only by a single glomerulus, which is a DA2 glomerulus. And what we are doing is we are exposing flies long-term for 4 days to geosmin, high concentration of geosmin, and then we study changes and modulations in the circuitry that's detecting this odor. And we see that there's a strong modulation going on at the level of local interneurons. We're also studying it at the behavioral level and can show that when flies have been exposed long-term to geosmin, then they are really tolerating it and they are not avoiding it anymore. So they just get used to it. And also they start then suddenly ovipositing on it. So the circuitry is changing at an anatomical level. And right now we are currently studying also the functional level to see now what impact does these anatomical changes have up the functional level, and what are then the consequences for the responses of the neurons. But the project I would like to show today is more on learning. So I briefly mentioned already that there's a valence code in the antennal lobe where certain glomeruli are encoding repellent odors and certain glomeruli are encoding attractive glomeruli, attractive odors. Now, this code is transformed and further pronounced in the higher brain centers, the lateral horn, and we could show some time ago that in the lateral horn we have a clearly pronounced valence code, meaning that there is a huge region which is coding attraction, so which is only coding odor attraction, a lower region here which is coding odor aversion, and a third region which is coding intensity in a way that when odors are presented or perceived in a high concentration, this region is being activated. So at the lateral horn, we think that odor specificity is more or less lost and odors are coded according to the behavioral value. Now we questioned what happens now when an odor, an attractive odor, for example, changes its valence. It changes suddenly its behavioral value to aversion. Does the code in the brain change accordingly? So what we did is to address these questions, we used aversive associative conditioning to change the valence of an odor. So what we do is we pair an odor with a negative reinforcer, which is an electric shock. So we use odor A, we pair it with a shock. We then present odor B, which is without any reinforcement. And afterwards we test, how does a fly chooses now, which odor does a fly now prefer? So we use the T-Maze, very well established assay by Joly many years ago. All the learning labs are using it. So we have the training part up here, where the electric shock is then induced to the flies, and afterwards the flies are then tested and can make the choice. So we use two attractive odors, acetylbutyrate, acetoinacetate, which we use at a concentration that they are equally attractive. So the flies, they distribute equally in the two arms between these two odors before conditioning. And when we then condition these odors and we pair, for example, acetoinacetate with an electric shock, we really shift the preference to acetylbutyrate. When we do it the other way around, we pair acetylbutyrate with a shock, we can clearly shift the attraction to acetoinacetate. So meaning, this shows we can switch the valence of odors. Now, does the switch induces any switch in the brain, especially in the lateral horn? What we did is we focused now on those neurons where we know that they have a clear valence code and that they mediate attraction, and these are multiglomerular projection neurons. So from the antennal lobe, two types of projection neurons convey the olfactory information to the higher centers. These are the uniglomerular projection neurons and the multiglomerular ones. The multiglomerular ones only target the lateral horn. They are inhibitory, they are GABAergic, and we showed some time ago that they really mediate attraction. Now what we did is we performed functional imaging by expressing GCAMP only in those neurons, which is the enhancer tripline MZ699. And when we do this and we perform now aversive olfactory conditioning under the imaging microscope, we observed that when we apply these two attractive odors, we get clear signals in the lateral horn and the multiglomerular projection neurons. But then when we measured the representation after training, we observed that the response to the learned odor is significantly decreased in the attraction domain. So the attraction domain is not as responsive anymore to this odor that has now switched to aversion. When we flip this around and now use the second odor and train the second odor, we see exactly the same. The activity to this odor is significantly decreased, while the control odor, which was presented without any reinforcer, has exactly the same response as before. So this shows there is modulation going on and that the attraction domain is significantly reduced in its response to an odor that has been reduced, that has been trained now and paired with an aversive reinforcer. Then we looked, of course, on the other response domains, and we observed that they are not modulated by learning. They show the same response before and after training. So only the attraction domain in the lateral horn seems to be affected and modulated by learning. Then we wondered, maybe it's just the electric shock, or maybe it's the long-term odor exposure that induces these effects. So we did unpaired training, which means we uncoupled the electric shock from the odor representation and then measured again before and after odor representation of two odors in the lateral horn. And we saw that there was no effect. So the responses were completely stable, showing that really the association is needed and the pairing of an electric shock with the odor is needed to induce the strong decrease in the response. Now we wondered, if we have this decrease in a response and these neurons are GABAergic, so they inhibit someone, the postsynaptic partner neurons should be equally affected. What are the postsynaptic partner neurons? These are third order neurons that innervate the lateral horn, so they get their input in the lateral horn and then they project to the ventrolateral protocerebral, and they are called VLPR neurons. So we focused next on those neurons because we know that they are inhibited by multiglomerular projection neurons. So what we did is we performed again functional imaging of these neurons, selecting a specific line that only labels those neurons and expressed GCAMP. And again, we did the protocol. One odor was trained with or paired with a shock, while a second odor was represented without any reinforcer. And then we measured the activity in the lateral horn of these third order neurons before and after training. And we observed that these neurons are indeed increased in their response for the learned odor, while the control odors are not modulated. So this shows that since these neurons are then probably less inhibited by multiglomerular projection neurons, which are decreased in the response after learning, we have an increase in the response of these third order neurons of the postsynaptic partner. Okay, next we wondered what might be the role.
Sorry.
Yes. What might be the role of these multiglomerular projection neurons for aversive associative learning? So what we did is now we manipulated these neurons, we silenced the output using the temperature sensitive Shibiri, and interestingly, we observed when we expressed Shibiri that, so I'm doing this a bit faster now, because I'm running out of time. When we express Shibiri, we observed that these flies still perfectly learn. So here in red, you see the activation of Shibiri. So we increase the temperature during training and during tests, but flies still can learn. We switch then to absolute conditioning. So next paradigm, because it could be that the Shibiri expression is maybe also changing the CS minus representation, but also for absolute conditioning, we again observe that these flies can learn. So it means these neurons are not required for aversive olfactory conditioning, although they are manipulated and modulated during learning. We then thought maybe these neurons are, since they are mediating attraction, maybe they are bidirectionally modulated and they also change after appetitive training. What we did next is we turned to an appetitive training protocol and paired an order with a sugar reward, so with a positive reinforcer. And we observed that these neurons indeed can be modulated in both directions, and they show a significant increased response after learning, again, only in the attraction domain. Lastly, we wondered whether these neurons are required for appetitive learning. So what we did again, we went back, we used Shibiri to block the output of these neurons. And this time we just blocked it during the test phase. So training was normal, the neurons were normally functioning. And during the test phase, we blocked the output of these neurons. And we could show while the parental controls perfectly learn, the flies where we block the output, they can't form an appetitive memory anymore. So this shows that these neurons are required for appetitive learning. Lastly, really my last slide, we also then wondered maybe those neurons can also generate an appetitive memory and they are sufficient to induce an appetitive memory. So we artificially activated those neurons using TRPA1. It's a temperature sensitive cation channel that we expressed in these neurons. And when you increase the temperature, you artificially excite those neurons. What we did is now we paired now an order, not with a reinforcer, but just with a temperature increase, so with an excitation of these neurons, and then tested the flies afterwards. And we observed that we can indeed, when we just activated the neurons, we could induce an appetitive memory, showing that these neurons, they are required, but also sufficient to induce an appetitive memory. Last slide just summarizes what I showed. So appetitive conditioning and aversive conditioning are bidirectionally modulating these neurons in a way that aversive conditioning is decreasing the response while appetitive conditioning is increasing it. This clearly affects the postsynaptic partner neurons, and we could show, where I didn't have time to show, that this is dependent on glutamate signaling and serotonin signaling in the antenna lobe. Last slide shows my group in Jena. So the work I just presented on the learning, it's a PhD thesis of Florencia Campitella. Here are all other members of my group. I showed a little bit the mixture thing from Ahmed and the mixture thing from Sudeshna. I didn't have time to go in detail into the other projects. Here are all my collaborative partners. As I said, I do most or all projects together with Bill Hansson, since he is hosting my group. So we have a lot of strong interactions. The learning project is also done in collaboration with Johannes Felsenberg. Here lower you see my funding agency, and thank you very much for your attention.
Thank you very much, Silke, for this really interesting talk. There are quite many questions in the Q&A panel, and also some people are putting the hands up. So I will try to start with a question in the Q&A. This. So there is a question from Johannes Felsenberg about the geosmin. Is there a critical phase for learning about the geosmin? And can you put adult flies on geosmin and see the same adjustment? Or do the flies have to be on it their whole life?
So there is a critical window, but this we didn't show now for geosmin, but I showed this while I was doing this during my postdoc with Leslie Fossall, and I showed this for carbon dioxide that only in young flies you can induce this shift, not in older flies. Young means they have to be between 1 and 6 days. And what was the second? I didn't get this. I didn't get the second part of the question.
The second part was, do the fly have to be the whole life in contact with geosmin?
No, so it doesn't need to be the whole life. It's just we do it for 4 days. And in the carbon dioxide study, I could show that even 2 days are sufficient to induce it, while 1 day is not sufficient. So, but from 2 days on already, you can change then the circuitry. Especially what we see on this, I forgot completely to mention, what we especially see is that the volume of the glomerulus that is responding to the odor, so the DA2, is strongly and dramatically increased. And this we see already after 2 days. And as I said, there's a critical window. You can only induce it in young flies.
Okay. Another question by Kevin Daly. Does the change in the inhibitory output of PNs in LH mean there is not a species-specific spatial map for odors? Also, can you observe in this assay if individual neurons drop out, or if the inhibitory PNs drop out, or if it is a general decrease in population response?
So, of course, we can't tell. I mean, we studied now mainly the inhibitory projection neurons. And of course, when we study the postsynaptic partner neurons, we don't know what other inputs they might get. And it could be that also other neurons maybe decrease in their response. We checked sensory neurons and we don't see anything. And we are planning now to check for the excitatory projection neurons. But this has been done by others already. So they looked on those neurons and couldn't find anything after aversive olfactory conditioning. So we assume that this decrease in the response is really specific for the multiglomerular projection neurons, but I can't be 100% sure.
Okay, thank you. So now maybe I will leave Annie ask his question.
Hi, Silke. Nice talk. So I have a question for also the geosmin story and the habituation. Have you looked for the expression of OR56A?
We have not, but that's a thing we wanted to do. But we haven't checked the OR expression. But I would assume, since there is a dream technique, and when you expose flies to long-term to an odor, you have usually a decrease in the expression of this receptor, right? So I would assume here the same, but we haven't done it.
Thank you. Zane, if you want to ask your question.
Hey, okay. Nice talk. Great stuff. Wow. Okay. So from your CO2 work with Leslie, so you showed that they're reversible in the sense that they increase the volume, you take them out, and they shrink the glomerular size. Is it true here?
Yes, we see exactly the same. We could confirm the former results that we have exactly the same. We have an increase in the glomerular size. What is new now is that we can really target single neurons. That I couldn't do before because it wasn't technically possible. Now we target single neurons and can really see that for local interneurons that we have more branches, we have more boutons, so that they are changing. While again, the sensory neurons seem to be completely stable and don't change. But we also see an increase for projection neurons, and also there we have a higher number of branches, so they also change accordingly.
Yeah, and a quick follow-up. So you're raising them on a single odorant. So just projecting into kind of holistic view of a fly's world, if you were to raise on a complex environment like a banana or a mole, what would happen, you think? Do they compensate at the end? Or how would the salience emerge from this complex environment?
Right, if you would put it now into nature and have, I would just say, so it is of course a very artificial situation that we're using, that it's just a single order, that its concentration is constant for 4 days. That would probably not happen in nature, that you have such a constant order exposure. And we just use this to understand whether there are any activity dependent changes. So in nature, I would assume, I mean, if a fly now grows up in a banana for 4 days, for example, also the larvae, that they usually, it could be shown that they are afterwards, that they also change their preference accordingly, so that there are changes later on, but usually then they go for it. They go for it when they have grown there in an early developmental stage, they would prefer this. And we see more that but we go for an aversive order, and we see that they are more tolerating it, which also makes sense because they lived on it and they just experience that you can very well live on it without dying from it. So then you can also just tolerate it. So I think it's really important that the olfactory system can adapt to these changes in the environment, because this is useful.
Thank you. I'm sorry, there are other questions in the chat and in the Q&A, but we are running out of time and we have to go on with the next speaker. Thank you again, Silke, for this very nice presentation.
Thank you very much, Emmanuelle.
So for the next speaker, we have a contributed presentation by Naoko Toshima, that is a postdoc fellow at Leibniz Institute for Neurobiology in Magdeburg in Germany. And he's going to talk about a rewarding and punishing effect of amino acid taste in larvae, in Drosophila larvae. Thank you, Naoko. It's your time. I hope you can share your screen. Good.
Hello.
Perfect. Thank you.
First of all, I thank organizers for giving me an opportunity to talk in such a great conference. I am a postdoc working in Bertram Gerber's lab in the Leibniz Institute for Neurobiology in Magdeburg, Germany. I am happy to talk about a rewarding and a punishing effect of amino acid taste in larval Drosophila. First, I love burgers. Probably I have eaten burgers hundreds of times in my life so far, and that has created an associative memory of a good burger taste and a burger smell. So if I was hungry and I felt a burger scent, then I would be attracted toward the burger shop. But if I have a burger in my hand already, then I have no reason to go to this burger shop anymore, and I ignore the smell. But this does not mean that I forgot the association between the taste and smell, but I just don't need to retrieve and behaviorally express the memory. So the expression of associative memory is dependent on your current situation and need. Similar thing happened to larval Drosophila. They don't go to burger shop, but they like sugar. After the training to associate sugar and a particular odor, they are attracted towards the odor. To be more precise, a group of larvae receives a presentation of an odor in a petri dish filled with fructose-containing agarose, followed by blank training without an odor and sugar. Another group of larvae receives an order presentation and sugar in the separated Petri dishes in an unpaired fashion. Immediately after training, an order preference is measured. Then performance index or a memory score is calculated from the differences between the preference of these two groups. And it is plotted like this. As you could expect, so here the appetitive memory is expressed as a positive performance index. On the other hand, if larvae are tested, these larvae received the same training as previous groups, but if they are tested in the presence of sugar, they do not show appetitive memory anymore because they have sugar around and they don't need to search for it. Now my question was, what about amino acid? Compared to sugar taste, an amino acid taste sensation, this mechanism is not well known yet. But amino acids are important nutrients because these are the building blocks of proteins, and in particular for larvae, they need a lot of them to grow. So I used a 20 canonical amino acid mixture instead of sugar. The training and testing procedure are the same as previously shown in the example of fructose. Here, as you expected, the larvae showed appetitive memory when they are tested on a pure Petri dish. But a surprising result came when I tested them in the presence of amino acid mixture. Now they are showing the negative learning score on amino acid Petri dish. This means aversive memory expression. To understand this data, I should now show you how punishment learning works in larvae. Beta substances, for example quinine, are commonly used as punishment in larvae. After training to associate an order and quinine, larvae escape from learned order, which is observed as a negative learning score in the presence of quinine. On the other hand, if larvae are tested in the absence of quinine, they do not show aversive memory, which is observed as 0 memory score here, because there's nothing to escape from. This is just like you don't always run into emergency exit unless you see fire or smoke in a building. Now coming back to my amino acid data. Here I'm showing rewarding fructose, punishing quinine, and amino acid mixture result all together. I think amino acid mixture is rewarding and punishing at the same time. When larvae are tested in the absence of amino acids, they show appetitive memory, but not in the presence of amino acid. But now, aversive memory, which was not expressed in the absence of amino acids, is now expressed in the presence of amino acids. This result also suggests that the larval brain is capable of forming two kinds of memory of opposite valences associated with one order at the same time. And either of memory is expressed depending on the current situation. Now a question arises whether distinct amino acids have either rewarding or punishing effect, or a single amino acid can have both effects. Or it could be that a particular combination of mixture gives such effect. To narrow down those hypotheses, I used all 10 essential amino acid mixture and 10 non-essential amino acid mixture and compared them to the 20 full amino acid mixture. In panel A, the larvae are tested on pure Petri dish to reveal appetitive memory expression. And in all three groups, the total concentration was same 10 millimolar. And here, in all three groups, appetitive memory expression was observed. On the other hand, in panel B, when larvae are tested in the presence of respective amino acid mixture, non-essential mixture did not induce aversive memory, but essential amino acid mixture did. This was a bit surprising because essential amino acids that animals has to eat are actually punishing to the animal. Now, essential amino acids has both rewarding and punishing effect. And so I tested individual single essential amino acids to know which of them has rewarding and or punishing effect. In panel A, larvae are tested on pure Petri dish to reveal appetitive memory. And it seemed that about a half of single amino acids are rewarding. And on the other hand, when I tested in the presence of amino acid, only tryptophan induced aversive memory. This means tryptophan can be rewarding and punishing. To confirm the effect of tryptophan, I used different concentrations. In panel A, larvae are tested on pure and B tested on tryptophan. In both cases, the memory score got higher when by the increasing concentration. The lowest concentration, 0.5 millimolar, is the tryptophan concentration included in the full amino acid mixture. Interestingly, this concentration did not induce aversive memory, which means that tryptophan alone cannot give aversive effect to the amino acid mixture. This also suggests the possibility that a particular combination of amino acids enhance the aversive effect of amino acid mixture. Now, as I said before, the mechanism how Drosophila detect amino acid taste is not well known yet. These papers 4 or 5 years ago showed that behaviorally and physiologically that the gustatory neurons expressing an ionotropic receptor IR76B are responsible for detecting amino acid taste both in larva and adult. I should note that the IR76B is broadly expressing in ionotropic receptor neurons, so this is predicted to work as a co-receptor, although the precise mechanism is not well known. So therefore, as next step, I used transgenic larvae to silence those IR76B positive neurons to see whether those neurons are also necessary for amino acid learning.
5 minutes left.
Here in this experiment, I used a 20 full amino acid mixture. In experimental group, IR76B positive neurons are silenced by the expression of potassium channel KIR. In panel A, larvae are tested on pure petri dish, and the experimental group showed impaired appetitive memory expression compared to genetic controls. On the other hand, in panel B, when larvae are tested in the presence of amino acids, the experimental larvae did not show, so the experimental larvae showed intact aversive memory score. This suggests that the appetitive and aversive memory formation for amino acids are independently regulated, and IR76B positive neurons are necessary only for the appetitive learning of amino acids. Now, currently, I am working on the same experiment for tryptophan memory. Let me summarize my talk. I found that an amino acid mixture induces both appetitive and aversive associative memory at the same time. I speculate that such a oppositely balanced memory might help for animals to find a food containing appropriate amount of amino acids, because it's shown that too much or too little protein leads to a prolonged developmental time for larvae. I also found that a single amino acid tryptophan can be rewarding and punishing at the very same concentration. Also, appetitive learning of amino acid taste might be mediated by the IR76B positive neurons, whereas we don't know yet for the aversive learning pathway. Yeah, I thank Bertram, the PI of our group, and all the lab members for the daily discussion. I received fellowship from the Uehara Memorial Foundation and JSPS, and we obtained the fly strains from Bloomington Stock Center and Dr. Richard Benton. Last but not least, thank you all for your kind attention.
Thank you very much. It's a very interesting story. So do we have some question? Please put your question in the Q&A chat or in the discussion. Let me share my screen. So, is there any hands up or any question for Naoko? I don't see.
There's one in the Q&A, Emmanuelle.
I don't see it. Yes. There is a question from Anna de Perty-Chauvin. For the adaptive memory, does the larvae immediately avoid the odor or first feed on the amino acids? And are they satiated?
Yeah, so we don't know how much they are eating while training or during the test. And what I can say is when the adult flies are used for such appetitive training, the adult flies are always starved. Otherwise, they don't learn about appetitive reinforcer. But when we use larvae,
Sorry, The question was about aversive memory. I'm sorry. The question was about aversive memory. Does the larvae immediately avoid the odor or first feed on the amino acid?
Yeah, so we don't know whether how much they are eating, and for satiety, and we don't starve larvae because we think larvae are always hungry, they are always eating. So when they train, we just take larvae from the food and just train, and they can show appetitive and or aversive memories. And yeah, how much they are eating, we don't know.
Okay, there is another question from Johannes Felsenberg. Could you elaborate if you think amino acids can act as reinforcement?
So the question means
What's the question?
yeah, so we think amino acid works as a reinforcer, and
it means as aversive reinforcement.
mm-hmm.
John is corrected.
Yes, I think so. So we don't know which taste modality the larvae is sensing amino acid. So it could be just simply bitter, or they have special sensor to sense the aversiveness of amino acid. We don't know that, but we think the amino acids somehow have aversive reinforcement effect.
Okay, thank you very much again for your talk. Interesting results. And now I will give the hosting back to Wynand. Thank you very much for the speaker, and let's go on with the next one.
Thank you very much, Emmanuelle, for hosting the previous 4 talks in such a wonderful way. So Naoko started the topic of taste today, and the next keynote speaker is Frederic Marion-Poll. So Dr. Frederic Marion-Poll is professor at the University Paris-Saclay in the Agro-Paritech Department of Life Sciences and Health, and also at the CNRS Department of Evolution, Genomes, Behavior and Ecology. So Fred has worked on olfaction in insects, first in honeybees with Claudine Masson, and later also with John Hildebrandt on moth olfaction. But he is most well known for his always insightful and thought-provoking research in Drosophila taste. So Fred's current research is about taste physiology and evolution in insects, with an interest on how to use taste ligands in crop protection. So it is a great pleasure to ask Fred to give his talk on bitter perspectives and insect taste. I cannot hear you, Fred.
I was muted, sorry.
No.
Now you see my screen?
It's coming, I think. Yes, very clear. Yeah.
Okay, good. Thank you very much, Wynand, and it's really exciting to be here. And I thank you, Walter, Wynand and Karl for organizing this really exciting meeting. We were missing meetings during these 2 years, and even this electronic communication is really beneficial. Today, I will not show you new results. I wanted to discuss with you. I wanted to ask for your help. And the reason is that, as Wynand said, I come from Olfaction, and I started to work on taste maybe a couple of years ago. And I find that taste is a mess as compared to olfaction. It's really complicated. And it's especially complicated if you want to use taste to in crop protection context. So what I want to do today is to explain what we could do and how I see the work we have done on taste, and then end up with a few propositions on what to do next. And I hope I will not push open doors too much for you. So my interest for taste and antioxidants came from the work of Greg Chapman, Liz Bernays, and John Glendinning. And I was really impressed by their ecological and physiological approach. And what they were interested in was to show that chemicals from plants were preventing insects from feeding on them. And what you see here in the center of the slide is a typical antioxidant test. You take disc of leaf and you treat 3 of them, and you see that this larvae discarded 3 of them. And Murray Eastman, from whom I took this picture, defines antioxidants as follows. He said that antioxidants are any substance that reduces consumption by an insect, and any such substance can be considered an antioxidant. And actually, there is a whole debate in the community between pure antioxidant effect and toxic effect, because you can stop feeding because it's bitter, or you stop feeding because you die or you are sick. So most of the people are interested in antioxidants because of their impact on the sensory system. But in short, an antioxidant is a substance that tastes bad to insects, and that's why we call this substance bitter substance. So actually, bitter substance in an agronomical context, people were hoping to use them as antioxidants to protect plants. And there is a whole body of literature where people propose to use antioxidants to protect plants, because if you use antioxidants, you use less toxic molecules in the environment, and actually you avoid killing insects so that they can be eaten by birds or entomophagic animals. But actually the use of antioxidants really failed, and people were disappointed by them, because the efficiency of antioxidants decreased with time and food deprivation. When I was a kid, my parents wanted to prevent me from eating my fingers, and they put bitter substance on it, and I became used to this bitter substance, and I continue to eat my fingers. Or if you don't want to eat salad, they told me you will have nothing else to eat, and then you are too hungry and you eat it. So the efficiency of antioxidants decreased with time and food deprivation. And the toxicity of these antioxidant compounds is actually very variable depending on the organism. And more than that, some antioxidants are stimulant for specialty insects, for example, gossip or cinnagreen. So these antioxidants are really complicated to use. But the thing is that many plants are producing antioxidants. I don't know any plant actually which do not produce antioxidant compounds. For us, we have selected plants, we have domesticated plants, and we have discarded the plants which were producing too bitter compounds. So we are used to eat plants which are not very bitter, but it's not the rule. And actually, you observe that when plants are attacked by insects, they mount defense, and the mounting of their defense include insecticides or defense, but also antiphedants. And these antiphedants are very diverse, and they belong to a lot of different chemical families like alkaloids, phenolics, and terpenoids. What you see on the right is something I liked. It's dandelion salad. I am from east of France, and we used to eat it when I was a kid. And what is remarkable with this salad, with the dandelion, that you can collect them and eat them as salad just before they flower. When they start flowering, they're extremely bitter, and you don't eat them. So the amount of antiphedant that plants are producing depends also on their stage. So if antiphedants are not useful in an agronomical context, why plants would produce so many antiphedants? So that's a question I cannot answer now, and maybe you have the response, but I don't know why they produce so many antiphedants. Maybe at the end I will make a proposition to answer this question. So what you see here is two pictures, two movies that were sent to me by Ayako Wada Katsumata. On the top you see a cockroach which was given sugar, and it's a happy cockroach. And then on the lower figure, you see a cockroach which was given a bitter substance, and you see that when it received the bitter substance, it really wants to get rid of it. So the perception of bitter substance induce usually a behavioral reaction, and most of the people who work on a bitter substance, they say that bitter perception prevents ingesting toxic food or foul food. And these reactions are innate, found in all animals, including in humans. The babies, they react to quinine quite readily. The detection of such bitter substance is done through an array of receptors. And yesterday, Montel showed you this slide with a whole array of different receptors. The most well-known are the so-called gustatory receptors in insects. In Drosophila, you have 68 proteins. It's also mediated by ionotropic receptors, by sodium channels, by trypt channels, and most recently Montel showed that opsins are also involved, as well as autopetrin. So these taste receptors detecting bitter substance are equipped with a whole array of receptors, and we will concentrate now on gustatory receptors, on GRs. And there is this beautiful paper from Linnea Weiss from Karl-Ernst's lab, where she was the first to show that taste neurons are co-expressing an array of gustatory receptors. It's very different from olfaction, where one neuron is expressing one receptor. In gustation, one neuron is expressing an array of receptors. And sugar-sensitive neuron, denoted S on this slide, they co-express GR5A, GR61A, etc. So they co-express a small number of gustatory receptors, but bitter sensitive neurons, they co-express up to 20 gustatory receptors at the same time. So that's a mess. I mean, if you want to characterize the way such a neuron is responding to substance, it's extremely difficult. We don't know how these receptors cooperate or interact or even fight each other. So, but the good thing is that all sugar-sensitive neurons, they express, most of them express GR5A, and most bitter-sensitive neurons express GR64A. So with these two gustatory receptors, coupled with the genetic tools we have available in Drosophila, you can activate sugar-sensitive neuron or activate bitter-sensitive neuron. And this is really a blessing. So one example of the use of this genetic construction is this. You have seen yesterday Caroline Reisenman shown that Drosophila, melanogaster, and others are repelled or deterred by L-canavanin. L-canavanin is an analog of L-arginine. And if it's incorporated in the protein, it's toxic. And actually, Drosophila melanogaster can avoid L-canavanin. And the fact is that if you kill or inactivate the GR68 cells, the flies are eating on sugar solution containing L-canavanin as if there would be no L-canavanin there. And the graph on the right shows you that a dose response curve in control groups and in the resulting construction, and the GR68 DTI construction, they don't detect L-canavanin at all. So it means that if you remove these bitter-sensitive neurons, you might be unable to detect bitter chemicals, and you die because you eat toxins. Actually, this is not completely true. We discovered also with students like Alice French, who gave a talk this morning, that some substance also inhibit the detection of appetitive substance like sugar. So, for example, sinigrin, if you kill the bitter-sensitive cell, the flies are still able to avoid feeding on sinigrin, and the reason is that sinigrin mixed with sugar makes sugar not sweet at all, depending on the dose. So insects, they have two systems. One system which is clear and that we know well, which is the bitter-sensitive neurons, and also sugar-sensitive neurons are able to reduce their activity. And it's possible that selection, evolution, makes these two systems really part of the adaptation of insects. Naturally, bitter-sensitive neurons respond also to other substances than antiphedans. With people in Dijon, we worked on the detection of 7-trichosan, which is a sexual pheromone in Drosophila, and we discovered that 7T is detected by, at least on the proboscis, by neurons which are also responsive to caffeine. And what you see here is a series of recordings showing that the cell responding to caffeine is the same as the cell responding to 7-trichosan. So in other terms, if you put caffeine on a female, the males are deterred. And if you put 7-trichosan on food, the males stop feeding as well. So the insects don't have so many neurons, sensory neurons, but they share the same circuitry. So depending on the context, a substance can have an influence on sexual intercourse or on feeding. We have also worked on chemicals which come from microbes, and for example, LPS or other compounds from bacteria are detected as aversive. And Craig Montel also showed beautifully that temperature is also activating some bitter-sensitive neurons. So bitter sensitivity should be called aversive sensitivity, but whatever, we call them bitter-sensitive neurons. So in order to sum up these ideas, we have two systems. One system which encode a positive valence, and it's the sugar-sensitive neuron which predicts if food will be nutritious or not. And you have a whole array, a whole range of gustatory receptor and neurons which predict if the food should be avoided. So what I find fascinating is that you have two groups of neurons which express distinct GRs. And I wonder how this partitioning between two types of two groups of gustatory ligands occur. Is it chemical nature of molecules or something else? We don't really know. So if you take a more evolutionary approach, you look at the distribution of bitter and sugar-sensitive receptors in different species, and you see that animals which are specialized tend to lose bitter neurons, bitter-sensitive neurons. For example, vampire bats. I know these are not insects, but they tend to lose their bitter receptor. They have access to food which has no toxin. And there was yesterday and today a whole discussion on Drosophila seychellia, which is adapted to toxic food. And yesterday, Danuka showed us that this adaptation to a toxic food occurs through the expression or the possibility to detoxify the toxin from the food, but also by a change at the level of the taste receptors, not only in the bitter receptor, but also in the sugar-sensitive receptor. And there's a number of examples also in the literature which shows the reverse, that some insects acquire bitter taste either because they are herbivores or because they specialize on a specific plant. So what you see here is Pieris rapae, which is adapted to cruciferous plants, and they have a receptor to cinegreen. And there's a beautiful paper from Wang and Al in 2021 describing this system where they showed that there's specialized receptor to cinegreen and a more generous receptor to cruciferous chemicals. So if you look more precisely at the number of receptors between in insect species, you see that specialist insects like human louse on the top of this table, they have very few taste receptors. Whereas animals which are polyphagous, like Noctuidae or Platella, they have up to 250 or almost 500 taste receptors. Can you imagine having 500 taste receptors? You live in an incredible sensory space. So there are exceptions in this general rule. For example, Apis mellifera is considered as polyphagous and has very few taste receptors, etc. But in general, it seems that insects, they have almost as many taste receptors as olfactory receptors. So this is quite amazing. Compared to humans, we have a very poor stock of gustatory receptors. We have about 30 receptors and 200 olfactory receptors. We are like the Apis mellifera. But these guys, they really have a huge number of bitter sensitive receptors. So what I didn't say is that when you have taste receptors, most receptors are devoted to detect bitter molecules, and only a few of them are devoted to the detection of appetitive compounds. So in Drosophila, for example, you have 9 sugar-sensitive receptors, and most of the others, 50 or whatever, they are devoted to detect bitter chemicals. What I find
5 minutes.
Sorry? 5 minutes, you said?
5 minutes, yes.
Okay. What is, during the discussion I had with Reich and Lisbon and Glandini, there is something that really I found really puzzling, is that specialist insects are more sensitive to antiphedons than polyphagous one. So there are two papers where they demonstrated that, but it was said on and on by electrophysiologists. And this doesn't match with the idea that polyphagous insects have more taste receptors. So anyway, so there is also this nice, beautiful paper from the group of Grosjean with the discovery of Jules Silverman that cockroach exposed to a toxin within a bait can adapt to these insecticides by learning, evolutionarily speaking, to avoid the bait, and they have a mutation that makes for them glucose as being a bitter substance. So this is crazy. Sugar, I thought that sugar was always appetitive, but insects are able to form gustatory receptors that makes sucrose as an appetitive taste, aversive taste. And they describe this very nicely in a science paper in 2013. So these dots that to the idea that insects may have that there is variability in the way gustatory receptors are expressed and in the sensitivity between individuals. So we need to develop tools or strategies to evaluate how much variability there is in taste receptors of insects. If we want to use them antiphedons in an agronomical context, we need to know how variable the response are. Otherwise, as soon as quinine would be on the market, the quinine will be overcome. So we need to know how fast this evolves. And so we need to do more population studies at the level of the behavior, at the level of electrophysiology, and also at the level of genomics. So in summary, taste and contact perception is an important sense from insects. The general architecture of the system is very different from olfaction. These gustatory receptors are surprisingly complex, and maybe insects are more discriminative, can discriminate more than we know now, and we need to develop an approach to estimate this variability. So where do we go from there? There is one problem, which is to identify the ligands of these receptors. If I were Grosjean and I had in front of me 486 gustatory receptors, I don't know what I would do. And there is another thing, is that taste is not only worse to change the behavior, it also changes the physiology. So we know this, we have seen this yesterday from the group of Christine Scott. So I make three propositions. One proposition that bitter being a predictor of toxicity is maybe not enough. Second proposition is that we may use bitter chemicals to protect bees. And the third proposition is that we may use bitter chemicals to delay resistance to insecticide. So bitterness predicts toxicity is not really something strong. The work of John Glendinning and Nissim et al., which shows that it's not really the case. Bitterness is not always inhibiting feeding. It's also a sensory cue. It may induce salivation, production of digestive enzymes. It may induce vomiting. And bitter might also be searched for. We have seen the work of, forgot his name, in PLOS genetics, that a bitter chemical can be a token stimulus, but it could be also searched for as a defense against microbes, parasites, or predators. We have a nice example in Drosophila where Drosophila is searching for alcohol because it helps them to defend against parasitic insects. But they may use chemicals like this to avoid predators or parasites. The second thing is comes from a work published by Geraldine Wright. She showed that imidacloprid mixed with sucrose is slightly more attractive, appetitive than sucrose alone. So why is that? I mean, the reason is that imidacloprid is new on an evolutionary scale, and bees have not learned to avoid imidacloprid and to associate a taste of imidacloprid with something that you should avoid. So if we would have been thinking about that, maybe we would have tried to design imidacloprid molecules which were bitter to honeybees, or associate a bitter substance to imidacloprid may protect bees from being intoxicated. So the third proposition is more speculative. In crop protection and in medicine also, we see that the most we use insecticide, the most we use a toxin, the quicker we select resistant insects. So this is mechanical. But if we would associate the bitter chemicals to an insecticide, let's take a population of insects. They are first confronted to the bitter substance, so a large proportion of them are getting away from the plant, away from the toxin. So it means that only a small proportion of insects are exposed to the toxin. So the result of this is that it will delay the apparition of resistance. So my proposition is that antiphedants, actually, they protect plants against apparition of resistance in plant. When you have a tool, when you have a toxin, you want to make it efficient as long as possible, and antiphedants may help to do that. So I'm finished with this. I thank all the people who worked with me during these past years, my collaborators, and really I want to give special thanks to Elizabeth Bernays, Ray Chapman, and John Glendini, because what I told you today comes from the discussion we had many years ago by email. And I thank the community of chemical ecologists. It was really great to see all of you today and yesterday. I was really happy to be with you. Thank you for your attention.
Thank you very much, Fred, for this very interesting talk with these interesting 3 propositions at the end. So I think John Pickett would like to make a comment on your main question. Yes, please, John.
Great, Frederic, and it was very good that you posed some serious questions there, because you know, we're so busy looking at the mechanisms and so on, and the chemistry and the neurophysiology, we sometimes forget the big picture, which is, can we protect food more sustainably? And you're obviously in a department that has to think about that. Your first question is really fundamental to many issues, and that is, why is it that so many plants produce antiphedants and yet they don't work? Well, it's really a question of adaptation, and of course all the main crop plants which we grow in abundance, which means there are very serious advantages for insects that adapt to them. They've all got their own antiphedants or toxicants, or antiphedants that at some higher level are toxicants, which is possibly to do with some antiphedants mechanism. The real problem is that there's no advantage in the antiphedant evolving to overcome the resistance of the adaptation. And it's not like a pheromone. If you have resistance to a pheromone, the insect's actually got to use another chemical for the pheromone, because it still needs to find the mate. And that's also true of host location semiochemicals. So there you can actually keep up with the resistance mechanism by simply identifying the next chemical that is replacing the semiochemical host attractant or the pheromone, and that's a rational process that we can do. But where it involves antefeedants, it's very difficult to have that situation, because you've by the adaptation, you've removed yourself from that situation. But you would need another plant where the antefeedant was of value. And that's where your idea of having the toxicant behind the bitter compound. In fact, it's a great idea, but it's been around quite a long time. A colleague of mine at Rothamsted, who sadly died of cancer before I could make use of him in his retirement days, when he could move away from rather dull insecticide resistance mechanisms, which was his main choice when he was paid to work. He really felt that we ought to combine antefeedants of various kinds, not just bitter kinds, with insecticides, so that there was a reinforcement when the antefeedant was overcome of the antefeedant effect, because they would then get the toxic effect. And he wanted to work on the dry main antefeedants to try and effect that. The real problem was we didn't have GM at the time. And so we couldn't put the antefeedant into a new situation where there wasn't already an adaptation. But we can now, but we would have to build into it this phenomenon that you refer to of a reinforcement of the toxicant by when the toxicant is engaged with, having overcome the antefeedant itself gets reinforced. But it's a sophisticated program, and it's a lot more expensive to advance it than just spraying with a broad spectrum eradicant insecticide like imidacloprid or clothianidin. But you're quite right. We have to think about this, and we have to think about the whole philosophy. And in fact, when I was thinking, I thought, what a great idea that we should meet after this Zoom today, this evening, those of us who are in a suitable time zone, with a glass of wine and kick these ideas around, because, you know, you need to have a few stimuli. Maybe we don't all drink, but, you know, we can have some sarsaparilla or whatever for those that don't. But I'm going to actually have to get on with preparing for my next talk, which is actually on the rational overcoming of resistance, not just to pheromones and semiochemicals that relate to host plant attractant, but also to insecticides and other pesticides, herbicides and fungicides, and also drugs. And I'm speaking at the American Chemical Society meeting in the US at the end of this month. Well, no, it's a week on Monday on this subject, and it's rivetingly exciting, and it offers prospects of rationally dealing with resistance to everything, including drugs. But you have an uphill battle on companies that are really the owners of these drugs, and really, I don't know whether they actually just want to keep selling new compounds that will be resisted, but they kind of resist any kind of rational approaches because it worked before. Yeah, that's it.
Yeah, thank you very much, John. Fred, do you want to respond to this now, or over the glass of wine later on?
No, I just want to add one word, is aposematism. The insects they communicate with, there are many insects or animals which convey the idea that they are toxic or dangerous to their predators using aposematic colors. So I think that bitter chemicals are aposematic molecules, at least some of them.
Right.
And usually we make a direct association with the toxin and the bitter taste, but it's not the correct thing. It's the bitter molecule is something that predicts the possibility of toxicity.
Thank you.
And if we think in terms of aposematism, then it makes us understand why insects might respond to bitter chemicals they have never met before. And it's in order for aposematism to work, it needs to convey a signal that can be easily understood by the predator. So that's one way to look.
Well, it's clear that there's a huge amount to discuss here, and wonderful ideas. And thank you for launching this discussion, Fred and John, for your very important comment. So I think we should move on at this point to the next talk, which we actually return to Olfaction here. And this will be Jonathan Bohbot. So he is in the Department of Entomology at Hebrew University of Jerusalem, and he studies the neurobiology of insect olfaction. So today he will talk about the evolution of indollergic odorant receptors, and this is a kind of talk that is related to the one that Jason Pitts gave earlier in the program in the first segment. Go ahead, Jonathan.
Hello, thank you, Wynand, for this introduction. I'd like to thank all the organizers for this wonderful meeting. I will launch the show function. So I'm going to tell you a story about indoles today. These are compounds I've learned to respect and discover more about as the years have passed. And until very recently, we were stuck with these indole sensitive receptors in mosquitoes. And it's only very recently that we were able to break out from mosquitoes. And this is a piece of the story here. So indoles are very ancient. This is a message I would like to convey. They are very ancient intercellular signals. They are the building blocks around which many hormones have been evolving in the different kingdoms, including plants and bacteria and animals. And you can see here on the screen a few examples of indole-based hormones such as auxins, serotonin, and melatonin. But they are not only used for intercellular signal, they're also used for inter-kingdom signaling. So this is an improved drawing of some of the interactions between species that are mediated by indole and indolic compounds. So you can see on the left, you have the plants, and you have some insects such as caterpillars that can prime the plant to signal to other plants that an herbivore is around. You also have examples of indole-based compounds that are repellent to butterflies. There are also components of flowers,
sorry,
mammals.
Jonathan, could you show the slide? You're not in slideshow mode.
Okay, thanks for your patience. Sorry about that.
So
you get the idea. So indoles are very common. They're ubiquitous compounds, and they're involved in inter-species communication. So we know some of the role of indoles in mosquitoes, in mosquito behavior and mosquito ecology. They've been traditionally ascribed to be oviposition site attractants, but as I mentioned before, they are ubiquitous. They are produced by the human body in great quantities thanks to our microbiota. The plants also have the biogenic ability to produce indoles. I have to mention scatol, which is 3-methyl-indole. It's an analog of indole. They're very close. And for some of you, you're familiar with the ability of mosquitoes to discriminate these two compounds, at least at the receptor level. So we discovered them in Aedes aegypti, Anopheles gambiae, Walter Leal in Walter Leal's lab with Culex quinque fasciatus as well. They usually share 50 to 70% amino acid sequence identity. This is quite significant considering the 20% amino acid sequence average for receptors within and between species. In Anopheles and Culicin, there seems to be a difference, a major difference. In Aedes aegypti, for instance, you have an additional scatol receptor, which is only expressed in the larvae. In Anopheles, you only have two of these receptors, OR2 and OR10, which are expressed both in the larvae and in the adult stage. And so our big question was, these molecules are very important. Obviously, many animals are sensitive to it, including C. elegans, bacteria, and humans. It cannot be only mosquitoes for which we have a mechanisms to identify indole, obviously. Could we find them outside mosquitoes? And they are a bit difficult to identify because the amino acid sequence identity between mosquitoes and flies is quite low. So we compared all the receptors from Drosophila with the receptors from Anopheles and Aedes and Culex sequences. And this is not really new. People knew that the closest homologs were OR30A, OR49B, and 43A in Drosophila, but they seem to be activated by other compounds other than indole and scatol. Their amino acid sequence avidity was quite low, between 26 and 40% within Drosophila, and with mosquitoes, the indollergic receptors around 32 to 36% amino acid sequence identity. Previously, using sensory physiology and the expression in heterologous expression systems such as the oocytes, aromatic compounds seem to be important in the activation of these receptors. Now, note that when these experiments were done, they used very high amount of these compounds, like 10 to the -2 dilutions. And when it came to the pharmacology, the EC50s, which is a measure of sensitivity of the receptor to the ligand, they were in the mid-macromolar range. So it's really significant amount of product that you're blasting into these receptors, and that's a limitation of these studies. Nonetheless, they were very important studies to start deorphanizing these receptors. So I will start with Drosophila melanogaster OR30A. This is a receptor that is expressed in the dorsal and the terminal organs of the larvae. That's specific to the larvae, a bit like OR9 is specific to the larvae in mosquitoes. We started our screen with a 10 micromolar concentration, and you can see you would expect that you would see a broadly tuned receptors at this concentration. Now, I need to be clear. We use a panel of indolic compounds. A few of these compounds were not indolics, and you can see a broad response of this receptor to different indolic compounds. But when you lower the concentration at 0.1 macromolar, 100 nanomolar, the tuning curve is much narrower. We took these different, the most potent ligands, and we established concentration response curves that you can see on the right. And you can see a very interesting pattern here. Now we're getting into the nanomolar range for indole, 3-indole acetic acid, and two others. And that is still puzzling us, and we can discuss this maybe briefly if I have time, despite all the technical problems I created. So here you have multiple ligands that activate this receptor in the nanomolar range. The next slide. Here, this is Drosophila melanogaster OR43A. It is selective to 3-methylindole and 1-methylindole. And here I would like to give a little thank you gesture to Marcus Stenzmer, who was actually, who gave us the impetus to start this study. So what we found out is that these two compounds are quite potent, maybe equipotent, you could argue. And what's interesting is 1-methylindole is not found in nature. Maybe it exists, I'm not sure, but it hasn't been characterized in nature. So here we're in the low macromolar range. And the last of the triad is Drosophila melanogaster OR49B. It is tuned to indole and also to 4-methylindole and 3 other compounds in the low macromolar range. So the other things that we were excited about is that with the help of
5 minutes.
Jason, 5 minutes. I will do it in 5 minutes. I'm not sure you'll understand everything I say with my French accent and the pace of my speech, but I'll try. So yesterday, some of you saw the presentation of Jason, and he showed very clearly that 2 receptors in Musca domestica, the common housefly, are very selective, one to 3-methylindole and the other one to indole. And this discrimination is very clear. And it's also the receptors that are sensitive into the nanomolar range. So we are seeing signatures of these receptors all over the dipteran clade. It is very clear in the case of Musca domestica that indoles are very potent ligands. We don't know yet if they are behaviorally significant. We suspect they are. And when it comes to Drosophila, based on my limited experience, I would predict that these homologs are indole-allergic receptors, but the correct indole compound has not been identified due to the low macromolar range potency and the fact that multiple ligands can activate these receptors. We are excited about these findings because after 10 years looking at these receptors, we finally were able to identify excellent candidates, homologs, in non-mosquito species. So I would like to thank the Israel Science Foundation for funding this research. My PhD student David Ruel conducted this research with the help of Yuri Weiner. And of course, this research would not be possible without our lab manager, Dr. Esther Yakir. I would like to thank Dr. Jason Pitts for his help. He's a very good friend, a great collaborator, and it's a pleasure to be able to continue working with him beyond his years at Vanderbilt University. And it's with great pleasure that I would like to acknowledge Dr. Marcus Stensmere for leading us to do this research. Thank you very much.
Thank you very much, Jonathan. That's really exciting, this group of chemicals, indole and its relations that affect so many different species of insects. So let's see if there are questions in the Q&A or in the chat. Raised hands. Well, maybe I could ask one question. Oh, Sharon. Sharon, would you like to ask a question?
Yeah, if it's okay. I love this story. I love the indole story. It's such a beautiful connection between those receptors. It's neat that you found the ones in Drosophila. Have you looked for structural similarities now that we have an idea about the 3D structure of the molecule? Do you think you're going to find signatures between the different receptors?
So the short answer is no, we haven't, but we have looked at motif predictions algorithms, and they're quite conserved. In the case, I think I could be mistaken, but in the case of OR30A, this is very clear that the conservation in terms of motif, protein motifs is very conserved. For the other two, you start to see divergences. But now that it is confirmed that ligand sensing subunits are similar in their structure with ORCO, I don't think how we could ignore and not do what you propose, Sharon.